Gamm, and I'm going to kind of follow JR's lead and stand over here. But I'm going to try and talk through a lot of my slides too. So hopefully it doesn't matter that I don't have his dangerous laser pointer, but I will use it just because I like to have. It's good for me to have something to do with my hands. Otherwise, I get in trouble. I'm going to start by just saying how fantastic that last talk was. I mean, you have no idea that. Yeah, you have no idea how advanced that was. I mean, I work as a pediatric ophthalmologist. We work with the Wisconsin Council for the Blind and Visually Impaired. They're wonderful there. I hear things here and there about what's coming up, what's on the horizon. But most folks they don't know. So that did wonders for me in my communication with my patients about especially when I'm dealing with kids. And so it can be a little bit depressing if you think of the world, and sometimes the parents are that way. Like you just take a snapshot of where things are now, and you can't see what the future is going to hold, right? So the hard part is trying to uh, project yourself 5, 10, 20 years from now, or project your child 5, 10, 20 years from now, and that is a very bright future. And because of folks like JR and what they're doing, and just technology in general building on one another. So I've learned a great deal, and I will ratchet up my optimism for the future for kids. And I also want to say that for years and years, being from Wisconsin, we have some urban areas, but a lot of rural areas. And so I've always advocated for when they talk about, okay, what should my child be doing? Where should I move them? So for instance, for monocular patients, I'm like, don't do soccer. I don't want you heading the basketball or well, I don't want you heading basketball and soccer, in which case you really got vision problems, but a soccer ball either. But I'll say, you might want to consider moving to an urban area because of public transportation. So as opposed to a rural area where you may be very, very limited and your world may be very small. But you're trading that problem then in for all the obstacles that you find and all the dynamics that you have in an urban environment, which I've never really considered until strongly until now. So again, the idea of doing this in an urban environment where we're telling our low vision patients to maybe gravitate to for employment opportunities and things is very important. And then the one thing I'll just throw this out there. So what I was thinking about was, so I just got back from a fishing trip in Ontario, and I got a 43-inch muskie. Anyone wants to see it, you just ask me. I'm bragging like all that. But the water levels were really, really different this year than in previous years. And so what am I blind to? I'm blind to the rocks under the water, right? So I can't see those, and I'm kind of talking to JR too. And this is nothing he hasn't heard before too. So what I use my GPS. So I'm looking to see where people who had my rental boat before me have gone. And so a year ago, they've got a track, right, going through all these little channels. And then this year I see the track veer into different areas, right? But I know they went there and I know it was safe, right? And they do it in real time because every time that boat is taken out, somebody, you turn on that tracker. And so I can follow somebody else's. Now, it's not going to tell me if there's a branch that's too low, it's going to knock me off my boat, but at least it gives you kind of a sense. And so with our cell phones, if 1 out of 20 people who aren't afraid of privacy problems turn it on and walk in real time and go around that pothole or go around that, can that be used to be able to say, oh, today I'm going to, make sure I make a left at or avoid this curb because everybody's avoiding this curb for some reason. So I wonder if that, just in terms of that real time sort of thing, I don't know. Yeah, yeah. Well, feel free. I just thought, when we were thinking about that, I just went back to my trepidation about trying to drive this boat and then slam into a rock that's somehow underneath the water. Okay, so I've totally thrown everybody off now because I've talked about fishing. So, all right, can we load up? Oh, here it is. Okay, so my title that I was given was tRNA, Genetic Engineering, and Stem Cells. And I added, oh my, and a picture of Dorothy, because when I got that, I'm like, that is an awful lot to cover in a relatively short amount of time. And they're kind of disparate topics. So this is a brand new topic. Put it almost every slide is brand new. So hold on to your butts on this one. So, okay, so my disclosures, very important to put up disclosures. JR did it as well. I'm doing it now. And the main reason is because you need to know where people's biases might lie. They could be very honest individuals, but we all have unconscious sort of biases that we have. And even though we don't mean to mislead people, we all have these things ingrained in us. And so you have to know where they're coming from. Example I would give would be if you go to a car dealer and you want to buy a car that they're selling or not selling, you know intrinsically, okay, I'll take it with a little bit of a grain of salt. But if you go to the car dealer and ask their opinion on boats, you're going to say, oh, this person knows vehicles, knows engines, and I'm going to take them, I'm going to take their opinion on boats really, really strongly because they don't have a horse in the race. Okay, so I have horses in the race, and so I work on stem cells. I have patents on the retinal tissues and organoids that are derived from human pluripotent stem cells. I've co-founded and I'm the chief scientific officer of a company, Opsis Therapeutics, that's based in Madison, that has support from Fujifilm in Tokyo, and now also with Bayer in Germany and Blue Rock. And I'm interested in advancing this technology for multiple different reasons. And I work with a lot of other companies to use it to advance their technologies. So I kind of want this stuff to be important. So take that. So you need to know that going into all of this, my talk. So I'm going to start with just a general genetics overview. A lot of folks are going to say, I know this already, but hopefully it can still help and orient you for the rest of the talk. Okay, so we start with a cell. You've got trillions of these in your body. They're all different. They do different things, have different functions. And for now, I'm just going to talk about everything in generic terms. And then within that cell, you have a nucleus. That nucleus has all of your DNA in it. Every cell has the same blueprint of DNA in it. Billions of individual little ones and zeros, blips that tell your body what to make and when, and in what cell. And that's all inside of the nucleus. So all that information is in that nucleus, but the things that it codes for, the things that it wants to make so that the cell functions appropriately, is made elsewhere. It's made in a different town. And so it has to send little copies of itself, tiny little copies of itself called RNA or mRNA or messenger RNA, that is basically a duplicate of that master plan that then goes off to the factories in the cell where it provides the mini blueprints for every single one of the proteins that your cell makes. And there's tens of thousands, if not more than that, of these individual proteins that go and then function within the cell and help it to do its job and stay alive. Okay, so that's if everything's going really, really swimmingly. But what if you have a mutation? So we all have mutations, by the way. Everybody walking at what makes everybody different. So everyone is different from everybody else. What we call a mutation really depends on whether or not it causes something we don't like, right? So if it causes something that we don't like, we give it a bad term. We say a mutation. And it can be anywhere within your DNA. And the most of your DNA is kind of a desert. We don't really know what it does, but a small percentage of it is the area that codes for these proteins that do function within our cells. And so if there's a mutation in that DNA which gets passed along in families, or can be just a brand new mutation that happens in an individual, those mutations are carried forward in all those little individual copies for each of the genes that go on to make the protein, right? So it's carried over from the master warehouse to the factories. And then those mutations cause problems or changes in the assembly of that protein, which can either cause dysfunction of the protein or basically remove the function altogether. And that can cause problems at first, but ultimately in a lot of cases, in the absence of a really critical protein, the entire cell will degenerate and die. So it gets disused and upset, feels neglected, and then degenerate. That's kind of the whole process. Okay, let's do this now with an idea towards choroideremia. And the idea, so for choroideremia, the cell, we really don't really understand exactly which cell is the main culprit or the main target for the mutation. Likely retinal pigment epithelium. And a lot of folks think that that's maybe the main one, if not the only one. Photoreceptors as well. We know they degenerate, whether or not that's because the retinal pigment epithelium degenerate first, and the photoreceptors need the RPE cells to function, and in the absence of it, because they're divas, and without their helper cells, they go on and die. And there's also the issue of the vascular cells that feed all of this too, and whether that has a role. But we'll say RPE and photoreceptors for the cell types that we're interested in. The DNA, the gene, is the CHM gene, okay? And CHM mRNA, that's the messenger RNA. That's the little truckloads of nucleic acids that go on to where the protein's being made. And then the protein is called Rep1. So that's a little confusing because it's nice if they all have the same name. So if it was CHM all the way through, but you make this little switch, and the protein is called rep1. Now, the function is interesting. We actually don't know that specifically. We know generally what it does. It's involved in transportation within the cell, but there's a ton of transportation that goes on in every cell of the body. So understanding what exactly is being transported when, where, and why is still something we need to understand. So I talked about earlier about technology and advancements in that. This is still really important because if you're trying to fix the function of rep1, it'd be nice to know whether you're talking about something that's being shipped by rail or by boat, because it might be a different approach that you would use. So, so the function is still a question mark. Okay, so moving forward. So this is then what I'm depicting is the CHM gene, so a long stretch of DNA that codes for ultimately for the rep1 protein. All genes have a start, so it tells the machinery when to start making the message and ultimately the protein, and when to stop. So it's not just one continuous strand of information. There are distinct starts and stops, and that'll be important for part of my talk. And then those starts and stops are also incorporated into the messenger RNA, and then that makes the rep1 protein. So the genetic mutations then, there are tons. So how can this go awry? Well, there's a lot of ways that this can go awry. So essentially any blips, changes, deletions, insertions, rearrangements in that structure of that blueprint can result in the rep1 protein being made incorrectly or not made at all. So I'm depicting a mutation here as just an orange bar randomly somewhere between that start and stop. That mutation is also carried forward in the mini copies that are sent off to the factories in the cell and then included in the protein. Now, if the protein is actually made but has glitches in it, then it's similar to, in this case, an analogy of a bicycle, but with square wheels. So it's made, but it's functionally irrelevant. It can't do its job. It's like a waste of time for the cell to even make that. If it's a big enough mutation, so if it's enough of a change in that blueprint, the cell will say, oh, to heck with it, and they won't even make the protein at all. So it won't even be there. Those are called loss of function mutations. There's a different type. It's called gain of function. We're not going to talk about that. They're dominant mutations. That's not what a choroideremia is. So it's more mutations that cause a lack of function or a lack of production of protein altogether. So topic number one, took them kind of out of order, is genetic engineering. So people in here are familiar with the clinical trials, which were very important, even though they didn't, give people back all of their vision or a lot of their vision. Still learned a tremendous amount from the gene replacement studies that were done and the clinical trials that were done. In that case, you don't even care about what's going on inside the nucleus. You don't care about the DNA. You're saying, okay, it's making a protein that doesn't do anything. So let's put our own copies, new copies of that into the cell and make the cell make new ones and bypass this whole system, give it a whole new gene. In genetic engineering, it's a different approach. You're saying, let's fix the gene that's already there. So we're not going to give you a new gene, kind of an artificial gene that will make that mRNA and that protein. We're going to fix the problem where it stands, which is inside the nucleus. And so that's essentially the idea of genetic engineering. And there are these proteins called editors. So, and a lot of them are derived from bacteria, but some very, very smart folks looked at this and said, they're in the world, in various critters and organisms, there have evolved ways in which changes in DNA can be specifically fixed. And not just randomly, but can zero in on certain parts of the DNA and say, you know what, that's wrong, let's fix it back to where it should be. And generally we call those editors. And they can be gene editors, they can be mRNA editors, they can do different amounts of correction, but they earned a Nobel Prize recently, so they've gotten a lot of attention. And so they actually fix that mutation in the DNA. So everything downstream of that would no longer have those mutations, the mRNA and the protein, presumably. Okay, then the editor goes away, mRNA is normal, protein's normal. Okay, fantastic. So why do we still have any diseases? So it's relatively new technology. These editors are big, so they're hard to package into the device, like the viruses and various other things that we use to get them into cells. Targeting the right cells is tough. They also are not perfect. There's a risk that in the process of fixing that mutation that you want fixed, that you introduce some random mutations elsewhere in your DNA. So you'll be trading choroideremia for leukemia, which we don't want to have happen. So all that has to be fixed or has to be worked out in terms of safety. The other main thing about it, just from a practical standpoint, is they're snowflakes. So as opposed to gene replacement therapy, where I don't care what your mutation is or how you got your choroideremia, you're going to get the same thing. I have got one product that I'm going to give you, which means that the manufacturing time, the cost, the regulations, the clinical trials, all of that can be done, and everybody can look at it with choroideremia and say, hey, did it work or not? Now, how young you are with it, what stage you are in the disease, that sort of thing all matter. But for the most part, you've got a one-size-fits-all therapeutic. With genetic engineering, it's mutation specific. So each one of them has to be specifically made for a mutation. And so when you have a disease that have dozens or hundreds of mutations, you're talking about maybe 100 therapeutics for choroideremia. Now try and do a clinical trial with that, right? So there's very fundamental problems that the FDA and scientists and clinicians are working through to say, how do we practically apply this so we're not making a $2 million product for every person walking the face of the earth, which is just not practical to do. So the sense of, and one of the things that we're working on and working with the FDA with is what's necessary for us to establish safety, and then if something works well with one mutation, how much do we have to do to prove that it will work well for mutations like that? Do we have to completely start from scratch again? Because that's not going to happen very often, most likely. There have been recently very good results, or decent results, I should say, not very good. So everything's relative. With one type of gene editing approach for retinitis pigmentosa, a form of Leber congenital amaurosis, where they use this kind of technology to fix a mutation in one type of retinitis pigmentosa. But it just so happens they pick that one because it has a mutation that's very common. So it covers most of the folks that have that disease, which is unusual in retinitis pigmentosa. They also picked a mutation that wasn't in the important coding region, but was in kind of in that desert region that I won't get into it, but leads to the disease. So there were a lot of things that were specifically kind of teed up for that first clinical trial. But it appeared to be safe, and in a certain number of patients that got it seemed to improve them. So there is a lot of promise for genetic engineering, but there's a lot of things we have to think through before it's just going to be boom, 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 moved into a bunch of different types of patients. But keep a close watch on that. Okay, so that's genetic engineering. So that applies to pretty much any type of mutation. So there are ways you can use genetic engineering to try to fix that. But as I said, each one is a snowflake, each one's expensive, and so it's hard to apply to a disease that has a lot of different types of mutations like choroideremia. So getting back a little bit to more broad-based approaches to therapies, you have the broadest, which is gene augmentation or gene replacement, the clinical trials that have already taken place. You have the most precise and custom and expensive, which is genetic engineering. And then in between, there are options that we have. And one of them are some approaches using tRNA, which I'll talk about next, for specifically nonsense mutations. So what is a nonsense mutation? It's a category of mutation where this normal stop site that I mentioned before there's a mutation that causes the signal to say you're done making the protein to be too far forward, to be in the middle somewhere. So it's cruising along. I'll show you in a little bit. I animated this. I'm really proud of it. I hope it worked. But it'll zoom along, and then it hits this mutation, and it falsely tells it that you're done. So it only makes a partial protein. So that's called a nonsense mutation because it makes a nonsense protein, okay, as opposed to a missense mutation like I showed you before. Okay, so in this case, that nonsense mutation, that premature stop in the coding sequence, is carried forward like everything else in the messenger RNA, and then causes there, theoretically at least, to be only a partial protein. In most cases, the cell recognizes that this is a fruitless venture and will degrade the messenger RNA and not even make the protein. Or if it makes the protein, it knows it's not good and just gets rid of it. Sort of a conservation of resources sort of thing, which is kind of cool on it by itself. So it's not essentially made. This is important because 21% of all CHM mutations are this category. It doesn't matter where it is. So it could be here, 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 here. It doesn't matter. So I'm not talking about a specific where it is, like with genetic engineering. I'm talking about what it does, which is a stop as opposed to a glitch. Okay, and that, although it's 21% of all the mutations, it's 35% or about a third of all patients who have choroideremia. Okay, so it's not the whole thing. It's not the whole enchilada. About a third. So if we could have a one size fits all for a third, that's pretty good. Kind of a meet you in the middle sort of thing. Okay, so how do they work? How do these engineered tRNAs work? So you have this so now I've eliminated the gene, and I'm going now to just the messenger RNA. So those are the little pieces of nucleic acids that go from the nucleus over to where the proteins and tell the proteins how to be made. So I've eliminated part of the cartoon that I gave you before. But as I said, that premature stop codon is still there, that mutation, that nonsense mutation. Okay, so this is a lot of work that we've done. This is not anything that I do, by the way. I'm also don't do genetic engineering, but I do stem cells that help advance these therapies. I'll talk about that at the end. So we work with Chris Ahern at the University of Iowa on this technology. It's really exciting, the technology I'm going to eventually get to here. And I want to just make a mention of his work because it's certainly not something that we do directly. We don't invent that. Okay, so that messenger RNA is, if we dig down into it, isn't just a generic piece of RNA. It has little bits of information that tell the cell to string together certain parts of a protein to make it a unique functioning entity within the cell. So those little parts are called amino acids. And so there are 20 different types of amino acids, and depending upon how you put them together in what order, everything will fold up a different way and ultimately make hopefully this functional protein. So I've depicted that here by overlaying this just blue line with different colored bars. Okay, and each different colored bar is a signal for the cell to put a specific Lego piece next to another Lego piece. Right. So when you're constructing the Rep1 protein or any protein, what happens is molecules called tRNAs or transfer RNAs recognize where you are in that coding sequence, and they bring along with them those Lego pieces, those amino acids. So in this case, you have a tRNA that's recognizing that, oh, there's a little yellow part here. So I'm going to bring along this yellow ball. And then after that, there's a blue one, and then I don't know what color that is, and then a purple one and a brown one. And so it marches along and adds the appropriate amino acid, the appropriate ball, and connects them together. Okay? And then as it's doing that, those balls have different conformations, and it causes them to twist right, twist left, and kind of fold up into that ultimately the protein that the cell is going to need. Now in this case, you have a nonsense mutation. It's bumped into that mutation that says, you know what, you're supposed to be done. Not true, it's lying, but that's what it tells it to do. So what happens is another type of molecule comes in there, binds there, and says, you're done, release. And so it releases, but it's only half made or 3/4 made or a third made, and the cell degrades it, and nothing ever gets actually fully formed. And so you don't have any Rep1 protein. Okay, let's go back. So there are types of drugs that have been around for a long time that confuse this step. And so what they do instead, they're called read-through drugs, is they coerce the cell to ignore those premature mutations and insert a random ball in there such that it will continue to move along. Okay, so it attaches this random ball, and then in a very inefficient way, it can proceed on its merry way all the way to the true end. The problem with that is that you're now trading a shortened protein with a full-length protein that has the wrong thing in the middle. So you still may be out of the frying pan and into the fire and still not making a correct protein. Also, it's not very efficient. And so what Chris and others have worked on to do is to make specialized, custom engineered tRNAs that recognize these mutations, but connect them not to a random Lego piece, but to the Lego piece that actually should be there to begin with. And it turns out they're more efficient and they make the correct full-length protein, such that when you get all the way done, it bypasses it, moves it along, and theoretically at least, you're left with a protein that's correctly made, correctly formed, and should do its function. Okay? That's engineered tRNA. And then it reaches the stop, let's go, and the world is happy. Well, happy face. Okay? So this is really new technology, and it's looking exceedingly promising, at least for that third of patients who have those nonsense mutations. All right. So we're done, right? So that we've got all this great technology. This we talked earlier about what's new. All of this I couldn't have talked about 5 years ago because it is relatively new and moving along at a very good pace. And there's a lot of excitement about it. Companies are being formed, and that puts more finances behind the effort, all of which is very good. And choroideremia is on everybody's kind of short list of things to apply this technology to, because there are so many individuals who have nonsense mutations, and because the Choroideremia Research Foundation is so good at mobilizing the community of folks that have choroideremia. So accessing patients, being able to say, hey, we've got something, where are you? That's a hard thing to do. And the companies will avoid diseases that they don't think they can get enough people to do a clinical trial for. So that benefit that the CRF provides is immeasurable. Okay, so we're not done though, because you can't just have a cool technology and go straight to a clinical trial, right? You have to establish safety. You have to be able to see, does it work? Does it work under circumstances that we think mimic the human condition? And for that, you need to have model systems, experimental model systems. So that's where you hear about mouse models and pig models and non-human primates. And now we do cell-based models. There's fish and flies. And you say, okay, well, that's kind of far away from human. That's true. But there are benefits to using animal models as well, that are far away from human animal model systems. So, and then now there's in silico. So being able to use computers to simulate things. So all these are being brought together to be able to try to determine whether your brand new spanking therapy is going to be safe and hopefully effective in human patients. Right, so we're going to concentrate on model systems. So all that I talked about before is now leading us into a discussion of what I do, which is stem cell therapy. And what we use stem cells for, and everyone thinks about it in terms of transplantation, and I'll talk about that at the end. But what I use it mostly for is validating or further investigating or refining therapies that need human cells and human DNA and human mRNA in order to understand how they work and whether or not they may help patients. So that brings us into stem cells. So kind of a stem cells 101. They're not all created equal. So when people talk about stem cells, oftentimes they just use it as a blanket term and everyone thinks, so they can do anything. There are bone marrow stem cells that have been around for decades and decades and have been transplanted into patients after irradiation and leukemia. They work great. They can make blood, and that's all they can make. So you can't take a blood marrow, your marrow stem cells, and put it in your eye and say, oh, it's a stem cell, so it's going to make eye cell. It doesn't do that. It's a bone marrow, it's a tissue-specific stem cell. It makes blood. That's what it makes. All it makes. So, but a lot of times when you go to the corner, shyster clinic, and they're doing stuff with stem cells, they're doing stuff with stuff that doesn't have any hope in Hades of ever making an actual photoreceptor or RPE. So they're not all created equal, but within the general category of stem cells, we have human pluripotent stem cells. And pluripotent is the key here. And that means that it has theoretically the ability to make more of itself forever. So it's an unlimited supply. It can make every cell type in the body. Okay, that's great, but it's also very difficult because how do you harness that potential to make the cell you want? So you're not making liver or bone, but you're actually making a photoreceptor or an RPE. That's hard to do. So the potential is great. Harnessing that safely is difficult. So there's two types. There are embryonic stem cells. Those were initially described in the late 90s at the University of Wisconsin in Madison by a colleague of mine, Jamie Thompson. And that led to additional research to understand what makes an embryonic stem cell an embryonic stem cell. And then in 2007, 2006, 2007, Jamie Thompson at UW-Madison, as well as Shinya Yamanaka in Japan, figured out 4 or so roughly genes that you could introduce into cells of our body, adults, that could revert them back to a state almost identical to embryonic stem cells. And because these are adult cells, skin or blood, that are induced to become embryonic-like, they're called induced pluripotent stem cells. And so this is what my laboratory works on mostly, is iPS cells. So why all the fuss? Why was everybody excited way back in the late 90s and the 2000s? Why is everybody disappointed now that we haven't cured all these diseases? Well, as I mentioned, unlimited supply. So it's a source issue. So if you have a disease, whether it be in your brain, your joints, your eye, where ultimately you lose parts, you lose cells like photoreceptors or RPE, once they're gone, gene therapy isn't going to help you because genes work inside cells. So if the cell isn't there, you have no place to deliver it to. So it's like replacing your furnace in your house before the house is unoccupied for so long that the whole thing falls to the ground. Delivering a brand new furnace to a dilapidated house isn't going to help you. It's not going to make it livable. If you get there early enough, when the furnace isn't working but everything else is doing okay, then that's fine. But late stage disease, where the degeneration has already occurred, that requires either bypassing the whole problem and going downstream of it or trying to replace it. But there was no source of human authentic cell types until pluripotent stem cells came along. So unlimited supply, potential source of authentic cells, but would they really be authentic? Or how close can we usher these cells all the way to something that's as unique and complicated as like a photoreceptor cell? No one knew. And then lastly, they're human. So that means they have human DNA, they have human mRNA, they have human proteins. And so all those therapeutics, the tRNA, the genetic engineering, all of that then can be tested because you have the background human DNA. Because even with non-human primates, there's enough differences in our DNA, thankfully, that you can't be assured that if you develop a therapeutic that's going to alter DNA in a human, that it wouldn't go someplace else or cause some other problems unless you actually test it in a human system. So all those things were what backed up all of the hype. So the question then is, can you take in this case a colony of induced pluripotent stem cells? So what I'm showing here is 2 kind of globs of cells. They don't look like much. They're just little gray dots, all kind of coalesced together on a culture plate about the size of my hand. And so can you take that and get them to somehow make a uniform, unlimited supply of retinal pigment epithelial cells? I'm showing right here, these kind of cobblestone looking cells that have a ton of functions that are specific for the eye. That's the only place they are. They have very precise jobs. Can you get them to not only make them to look like them, but actually function like them? And this is what they look like for real. These are real human RPE cells. And can you get them to make relevant to choroideremia photoreceptors? And so they are even more kind of cool looking, which is not good for when you're trying to make something. The more complex it is, the harder it is to make. So what I'm showing here in red is from a friend of mine, Nico Cuenca in Spain, who's a fantastic histologist. And this is what pictures of in this case, cone photoreceptors look like. They have little antennae that detect light. They have a kind of a power station that generates a lot of energy. They have a nucleus, a cell body. They have a wire that leads to a synapse that connects to the next cell. Each one of those has thousands of very precise parts that have to work in concert for the whole cell to function. So can you go from that little gray nothing to something as specific as a photoreceptor cell or an RPE? And that's where we were about 15, 20 years ago. What I'm going to tell you is yes, we can. Really close. Can't be 100%, but we're getting very, very close. So what do we start with? We start with human patients, most often, whether they have a particular genetic disease or not. We usually take a blood sample like you might submit for a cholesterol check, although we do take skin biopsies still. And then we grow those, we reprogram them and grow them in these culture well plates that are about the size of my hand, at least, a little bigger than my hand. And then inside of that, here's one of those kind of nondescript colonies filled with little iPS cells. Okay, those iPS cells are then, over the course of the last 15 years, we've developed ways, and other laboratories have helped, and now there's a lot of different technologies to be able to get from point A to point Z, but we manipulate them in different ways. We add different chemicals and cues to them. We physically manipulate them in different ways by making them in suspension cultures or 3 dimensions, plating them back down. There's a lot of different ways physically and chemically to adjust them, but what we're doing is we're trying to convince them to follow a developmental pathway towards becoming retina. Not liver, not your right pinky toe, but retina. And so that took a long time. And so one of the first steps is to make these into round balls that float around in the dish, because as it turns out, anything in your central nervous system, which includes your retina, likes to grow in 3 dimensions and likes to grow non-adhered, so not tightly bound to something, which makes it hard to handle. And a lot of folks were just trying to make sure everything's stuck down because it was easier to see and to handle. But that was one of the critical things early on that we learned, is that we had to give up that kind of safety blanket of keeping it in one place in order for them to really fulfill their mission ultimately to become neural. We then plate them back down. They form these little neural tubes, just like we would see with human development. It's really quite incredible how much you can recapitulate in a dish. We then pick them back up again, and they make these little golden Cheerios, these 3-dimensional objects that are really easy to see, and they look like developing what we call optic vesicles. And those are the little outpouchings that ultimately become the retina during normal human retinal development. So how do we know those are retina, primitive retina, and that they'll become something that's more mature? So we could do that by using various antibodies and other tests to define what types of cells they are. And so right off the bat, early, it's about 20 days or so into the culture system, we can tell that they're expressing all of the right things for a primitive retinal cell. So a cell that hasn't decided to be a photoreceptor or an RPE or anything else yet, but it knows it wants to be retina when it grows up. So it's kind of a high schooler. Doesn't know exactly what it wants to be, but it knows it wants to be like an engineer, but could change his mind, doesn't know, and then we follow that over time. And so once we get the freighter out of the port, it's remarkable how little we have to do. It's like once it knows what it wants to do, you have to do a lot of work up front, and then it's a waiting game, because it will follow a time-based sequence that it kind of just knows how to do. And we're still trying to understand all the steps that go into that. But we're fortunate that we don't have to micromanage every step along the way, because it takes 150, 200 days, similar to how long it would take to make a retina in utero. And so if we had to micromanage every step, that would be very hard to do. So instead, it kind of, once you get it going, it kind of knows what to do. And so here we start to make, after about 30 to 40 days, some primitive photoreceptors shown in red and yellow. So it goes from not knowing what it wants to do to the cell starting to say, hey, yeah, I'd like to be a photoreceptor. And so they have specific, express specific proteins that tell us it's kind of taken the first step towards being a photoreceptor. This is work we've done recently and published recently with our UW collaborators, Tim Gomez and Sarah Rempel. This is time-lapse imaging of one of these organoids. Each one of these black areas is a photoreceptor. And you can see how, this is short, but you can see how they're kind of moving back and forth. This is after 40 days culturing them. And then after 60 days, you can see now there's more of them. They're moving towards the outside. They're starting to make a layer. And then after 80 days, see this thick layer of photoreceptors. And for those of you who can see, there's these little wires that are working their way down to contact the next cell type that they need to contact. So you're starting to make them. There's only a few, then more and more get made, and then they migrate and they start to make a retinal tissue. So they're starting to make a photoreceptor layer, which is what we wanted to see. That was very exciting. After about 200 days, not only do they still look like these little balls, they've gotten quite big now, but big is relative. This is like a really big pinhead. And then we start to see on the surface these hair-like extensions. Those hair-like extensions are outer segments, which are the antennae that detect light. And that's one of the most, from a structural standpoint, one of the most definitive parts of a photoreceptor and one of the most complex. And so on the right here is a blown up picture of just the edge of one of these, okay? And in red are cone photoreceptors. This is all from IPS. Remember, this started with somebody's blood sample. And so each one of these red cells is a cone cell. These green blobs are bipolar cells, which they're supposed to connect to. And you can see the wires coming down and touching them. And I'm going to start this movie here. Oh, no, let's go back. Let's see here. I'm going to run, make sure I don't trip over anything. I'm going to get this going. Oh, no. Go back. I had a cool movie. So, okay, something doesn't work. But, oh, there it is. Something's up here. All right. Well, it's not moving. Anyway, all right. Spent too much time on that, I think. But all right. So as I said, so these are cones. This yellow one right here is a newborn rod. Rods are born a lot later. And so, but I want you to concentrate on this. So if we go, there we go. So here's just looking at those cone photoreceptors. And now let's compare it to real live human photoreceptors. So not perfect, obviously, but we have the little areas that collect light. This is all in a dish. This is in an actual human eyeball, right? So in an actual human eyeball, you have the little antennas. They're there. We have the cell bodies, which are there. We have the energy compartments, which are there. And then we have the connectors, which are there too. So really quite remarkable how close you can get to an actual human photoreceptor, which is arguably one of the most complex cell types in the entire body. So that's great, but that's wooden duck versus real duck. Do they actually function? And so this is work that we published this year with more collaborators who do electrophysiology, who actually expose monkey retinas and other things to light and then measure their response to wavelength. So does it respond to red, blue, green? Does it respond to different background illuminances? Does it respond more when you shine more light on it? And it turns out, yes, it does. And so the red cones respond to red light, the blue cones respond to blue light, the green cones respond to green light. And in many cases, they respond just as well as non-human primate or monkey fovea, the very center of the vision. So it was one of the first times we were able to show that not only do they have all the bells and whistles necessary to be a photoreceptor, but they actually could function as well as one that was in a non-human primate. Okay, so that's all photoreceptors, but as I said before, the seat of the disease may very well be RPE. That's what most people think. Turns out RPE is easier to make than photoreceptors, and people have been doing that for a long time. And so this is a hole where one of the retinal organoids was taken out from. And if we follow that and treat that a certain way over time, we can develop that into retinal pigment epithelium. And they also harbor much, if not all, of the function of real RPE as well. Okay, so what do we do with all this now? So that's just a lot of background. The fact that what I'm trying to tell you is that we can make iPS cells from essentially any living human being, and actually don't have to be living, if we get the samples, and then we can make the appropriate cell types relevant to choroideremia with them. So have we done this with folks that actually have choroideremia? And the answer is yes, thanks to Kim Edwards, who is a graduate student in my lab and was the first Randy Wheelock Award winner. And so she has made actually multiple tools, iPS-based tools, that both to just study Rep1 as a normal protein so we understand it better, but also that have a bunch of different relevant mutations that cause choroideremia. And so this is just 3 of the lines, and as I just want to show you, because this is unpublished, and since we're broadcasting this, I can't give you all the details, but I'm happy to talk about people and talk to people, and so will Kim. But they make these beautiful organoids. They have the hairs on the side on them. They have the antenna. They have all the parts to it that I showed you before. So it's a great resource, and in this case, they also have within them the mutations that cause choroideremia. And she's also been able to make all the retinal pigment epithelium. This is a gray-white image, so it doesn't look brown like the previous ones, but they also make the relevant cell type. So we now have a battery, a toolkit of iPS cells with multiple different types of mutations that we're really looking at to understand what it is that causes the disease to occur. Why is it only in the eye and not elsewhere? I mean, the gene and the protein's in a lot of cells in our body, and they're all perfectly normal. So why is it what makes RPE and photoreceptors special when it comes to choroideremia? Those are all things that we're interested in understanding, and we think that that will lead to better therapeutics and better readouts for clinical trials. Okay, that's fine. I mentioned before, and we're getting towards the end here, but I mentioned before how if we're modeling the disease in a dish, we're not intending to put that back into a patient. We're using that then to say to company A, B, or an academic, let's take your therapeutic, put it into those cells and see if it fixes things. So that's good for patients who are earlier on in their disease when the cells haven't died. But in choroideremia, over a period of time, those cell types, RPE and photoreceptors, eventually do degrade, in which case just modeling the disease isn't going to help. The cells are gone, so why would you model the cell? So we want to help, we want to have a system where we can help folks that are in later stages of their disease as well. And so we concentrate on cell replacement, so transplantation of those iPS-derived RPE and photoreceptors. So not only can we make them in a dish to help model the disease, understand it better, test these genetic engineering approaches and tRNAs, but can we then turn around and put them back into a patient who needs new ones? Okay, so the answer to that is we worked very to great lengths, not just to be able to show you what I did earlier, but then to say, can we convert that to something that is can be made safe for humans and scale it up such that you can make billions and billions of them? Yeah. Okay, not going to make it. So I'm going to go like. And we're able to make them in such a way that they are sterile, reproducible, and so that's something that can be achieved. So the manufacture of these spare parts have gone from artisan breweries to Budweiser, okay? And it can be done. It took a lot of effort, took 5 years, took about 24 people working around the clock for 5 years to do it, but that was possible. And that's key because you have to be able to, in order to work with companies and bring this forward, you have to be able to show them that you can make enough so that you can treat people for many, many years. So no one's going to do it and then have 5 years of treatment and then have to start all over again with a new clinical trial. On the right, then that's photoreceptors.
If that bag is not very dark, you're taking everything out.
You did. Eric is 2456. I saying for the
large kind of hanging IV bags of RPE. So both of them can be scaled up and manufactured for large-scale worldwide use. So that's great, but what about installation? That's, as I mentioned earlier, that's still a challenge. So making the product is important, but getting it into the appropriate location and getting it to shake hands with the cells that are remaining is still an issue. And there is a point at which we're not talking about just degeneration of the photoreceptors of the RPE, but what is the state of the rest of the retina? So if you've just made new spark plugs and you throw it into an engine and the engine's totally seized and rusted out, not going to help you too much. So it's not a cure-all, it's not a panacea. So even in the best case scenario, even if it was able to hook up, there's still a window where some individuals are going to be too far gone for that, and they need a whole new retina. They don't just need 1 cell or 2 cells, they need a lot more than that. So it's important to see that nothing is a silver bullet for everything, and we're trying to understand that so that we pick the right patients to be able to treat. So a lot of questions that we're working on now. One is, if we're going to extract those photoreceptors and RPE from their environments, from the culture environment, are they able to reach out and find new targets? And this was again back to our collaborators at UW-Madison. And the answer is we found out that there's a window at which those photoreceptors have the capability of sending wires out. So this is a photoreceptor made from an iPS cell, a human iPS cell, plated, and you can see it has what's called an axon. And this is the wire that's looking to make a connection with somebody else. And there's no somebody else here, but it's looking in vain. And so that sort of dynamic search that we're seeing, we see also in other neurons in the central nervous system. And if it doesn't have that, it's basically not looking for love, right? So there's a window where it is, and we want to be able to understand that so that we're not putting really cool looking cells that no longer care about, having a partner, but rather cells that are plastic enough that have the ability to form those connections and then maybe mature as they sit within a person's retina. And then how do we deliver them? So it's good to know that we can make them, good to know what the window is that may be best to transplant them, but then how do we safely get them into that delicate space beneath the retina? And so for that, we've worked with other collaborators at UW, Xiaoxin Gong and Zhencheng Ma, who are unbelievable engineers. And this goes back to when I say engineering always wins in a fair fight with biology. And so what they do is they make materials and scaffolds. And what Jack Shencheng does mostly is to make computer chips. And he makes the tiniest things in the tiniest spaces very reproducibly. And I said to him, okay, can you do what you do with microchips, but do it with a biodegradable scaffold that I can stuff cells in? And he goes, that sounds cool. All right, I'll try. And his wife, Sarah Xiaoxin, makes materials that biodegrade in the body safely. And so I said, can you make something that will be flexible enough that will fit next to a retina so it doesn't erode and will biodegrade? And she said, okay, let's try and do that. And so we ended up with a lot of different designs. This actually isn't our current design. It's a kind of second generation. We have a third generation now. Kind of call it our ice cube tray design. And it looks like an ice cube. If you actually look at it with the naked eye, that's what it looks like. You can't see any of these features. It's really, really thin, really tiny, but actually pretty manipulable. So a surgeon can load it up into a device and get it into a retina. So all those properties that were hard to work out. We can line these things with retinal pigment epithelium. So the RPE, if we're going to try to replace both RPE and photoreceptors, which is what we're going to have to do with choroideremia, then we have to be able to to make a double layer sandwich and get them to stay together and get into the space. And so each one of these red kind of polygons is the outline of a human RPE cell, and they'll line that entire structure. And then there's still a little divot there that we can put photoreceptors. Every one of these red globs is about 20 to 40 photoreceptors that are sitting in that pocket where the RPE is. And then we can get that into the subretinal space. And this is work that we've done in collaboration with the National Eye Institute and Kapil Bharti, who's involved in CRF as well, and Juan Amaral, who is a pig subretinal surgeon, Arvidas Mamanishkas, who makes a lot of the technology for this. And so what I'm showing you here, this one I did want to work and it's not working, unfortunately. So, and I don't think it works over there. This is an Apple thing. I don't know if you can you toggle over this? And what'll happen is you'll see maybe you can do that. I don't know. If he doesn't, I'll walk you through it. So here's a retinotomy. So here's a retina. This is a pig. So here's the back of a pig's eye. This is the pig retina. They've already made an incision. Down there. See if it goes. Yeah, it didn't work for me earlier. I don't know why that is. This is an incision in the retina, and this is a custom device. You can't see it because it's transparent, but a scaffold that's oval shaped is right here. And then if you could, you'd see that it would be injected into this space where it goes under the retina and it sits right here. And then that's lasered shut. And then they fill it with an air bubble. And again, I can't show you the anatomic and other results because they're not published yet, but it does a really good job of delivering those 2 layers of cells precisely to where we want to deliver them. What's the downside of it? You're restricted in size, right? So this would be good for somebody who's late stage choroideremia who would like to have a smaller area of vision back, but you're not going to be able to treat all the periphery, right? So this isn't something that you're going to be able to completely restore vision with, or again, limited in terms of completely restore, but that you're not going to be able to improve vision all over the place. It's going to be discrete areas. So there's always pluses and minuses to these technologies. And then lastly, how many questions are there? That's question 3. There's a lot of them and a ton that I haven't even addressed, and I'm happy to answer any of them. And then I want to just finish by going back to take a 1,000-foot view, because I talk to patients all the time about clinical trials and the frustration about how long things take. And I hope I've been able to convey to you just how many difficulties there are along the way and how many things that have to be worked out to make sure that we first do no harm. And so I'm showing here switchback roads down a mountainside, and kind of the idea being that every step along the way, you have to have an idea, you have to have an animal, you have to have a model system to test it in, you have to have funding for it, you have to do the initial trials, and then you do the late stage trials, and so on and so forth, and finally you get to the bottom of this mountain. And the analogy that I like to use, because it has to do with cheese, is if you cut that out, so you'd have what would be equivalent to the Cooper's Hill cheese rolling race, which is in Gloucester, in England. I don't know if you ever heard about this, but okay, so they take a wheel of cheese and they send it down this really steep hill, and thousands of idiots start chasing after this thing, and apparently there's one guy who's really good. He's like half the time he gets the cheese, and your reward is you get the cheese. But tons of people break their necks, right? And so if you barrel down a mountainside and ignore all the steps along the way, you may get lucky and you may get the wheel of cheese, but the vast majority of people are going to get hurt. So these checks and balances along the way, while they're not perfect, are still important. And the eye is leading the way for all the reasons I talked about before. So we're on the forefront. So we have to even be more careful about breaking our necks, because it's the people with eye disease that are going down that mountain first, right? And everybody else is watching us. So we want to make sure that we don't do anything that's reckless, because it'll set everything back decades, just like gene replacement or gene augmentation therapy did. The first ones weren't reckless. People did what they thought was right, but they caused leukemia. Some people died, and it was put on hold for decades until Gene Bennett and others brought it back very safely. So if people are too quick to try and grab the brass ring and they do something wrong, then everybody shuts down. The companies don't want to invest in it. FDA gets skittish, and that's it for progress. So I wouldn't say we don't want a hare, we don't want a tortoise, we want a really fast tortoise. We want the fastest tortoise we possibly can get. And then we want to manage expectations, because, as I alluded to, you didn't get from inventing a computer to having one on your wrist, a supercomputer on your wrist, overnight. You had big clunky things that looked like they had promise, and over time the technology improved and you had different iterations. Same thing with everything else. This is going to take time. The initial therapies hopefully have some signal, they look good, and they allow us to go back in, make them better, and over time will improve, have better outcomes. But it does take time to do that. And along the way, people are always looking to take money off of you or put you in harm's way. There's tons of snake oil out there, people who have conflicts of interest, who are charging you money. And what I generally tell folks is, if you're putting down your own hard-earned money on a therapy, run away. Because we're at a stage now where we have to ask you nicely to be involved. We have to run through all the risks for you. We're paying for you to fly and be involved in a clinical trial, because you're so important. We're not asking you to pay for anything. So a quick rule of thumb is, if you're doing something that they're taking your money for, they have a conflict of interest, and be very, very wary, or at least ask somebody. And these are the people who do all the work. I pretty much do nothing. I put together PowerPoints, and but I'm good at it, I think, at least. And then all of our funding sources. And most importantly, I want to thank the Choroideremia Research Foundation, who I really would. There's a lot of different ways labs like mine can go, and sometimes it's just a matter of who shows interest, the connections that you can make along the way, and people who influence your life. And in that regard, Randy Wheelock, I want to say, he's why I work on choroideremia. I was happy to work on choroideremia. I have lots of patients who have it. It's a disease that have lots of questions, and I'm intrigued by questions. But the impetus to say this and not maybe 20 other different things was Randy and his story. And so I'm eternally grateful for him. I did not know this young guy at all. So, but you'd probably be happy I'd be showing a very young picture of him. But so I want to thank the CRF, and I want to thank Randy. So thank you very much.
Thanks to both Drs. Gamm and Rizzo for such incredible presentations. Just be mindful of time. We've got, we're going to have lunch, and that's going to end at 1. So I think what we're going to do is we're going to have a few minutes for questions. Clearly after, and if you need to leave now, please do so. And clearly Doctors Rizzo and Gamm will be here for a little bit longer, but we're going to probably take questions for another 10 or 15 minutes or so. And then we'll officially break for lunch, but again, we can still be here for a little bit of time. I just like to say that my first experience with this conference was in 2018. And when you think about it, everything that we just saw for the last hour and a half, except for the historical information, but almost everything that we saw has just come around in the last 5 years. And so I think that that's pretty amazing. And we'd just like to point that out. And so with that, hands up, and we'll bring some microphones around for right behind you. You got one right behind. I'll come right back to you.
Hello, is this on? Oh, for Dr. Rizzo, just real quickly, would you consider this a replacement or perhaps a technological advancement on the guide dog?
Here, maybe better for a microphone, right? Oh. Oh. Whatever's easiest. Oh, okay, you can hold on to that then. Okay, great, we're live. It's a great question. I think the guide dog provides tremendous value, as does the white cane. And everything I presented today, I didn't really have time, but I just wanted to reiterate the importance of primary mobility solutions. They're super important, and you should always use them, right? All of these everything I presented in terms of the wearables and all the new services that we're trying to provide are supplemental, secondary solutions that should be layered on top, right? We should always have a first defense, and everything else should be considered a secondary defense. So, do I consider it to have a lot of the similar functionality to what a guide dog could provide? Yes. And do I consider it to, put ourselves in a situation where we could continue to provide service updates and improvements? Absolutely. And I think that's where the future is going. I mean, if we look to Tesla as a model system, if you will, not necessarily animal model systems, which is what we're hearing a lot about, but as kind of an engineering model system, right? Tesla has done a marvelous job. Although I don't always agree with Elon Musk's statements in terms of really misleading the public at large by saying he was going to put a driverless car with SAE levels 4 or 5 on the road years ago, and he really hasn't delivered on that. But in really pitching this enhanced autopilot, right, and then coming up with iteration after iteration where he continues to improve through over-air updates, which is really a marvelous model. I think we can really position ourselves with new assistive technologies that don't have to be static, but that really live and grow with you. And so, I don't really want to get into a situation where we're kind of setting one solution against another. Really, this is an enhancement. And I just want to give a quick shout out to all the amazing work that orientation and mobility instructors provide for us. A lot of the work, and you probably saw those names are really small. This has all been developed with orientation and mobility instructors on board the entire way. And I really think that's critical to any platform that's developed.
There we go. First of all, I want to thank both of you for fantastic presentations. Hearing you speak is just humbling. So it points out how naive and foolish and ignorant I really am. This is for Dr. Gamm. In the process, vision is only ultimately achieved if the the one side of it is the reception of information, right? Which it sounds like what you presented is achieving. The other side is the interpretation of that information from the brain. Is there any fear that or any work that's been done to say, hey, what we've produced in the photoreceptors is actually producing the right information, so the brain receives that in a way that it can interpret it equally as well as a normal retina?
Great question. I think therein lies a bigger discussion about different types of intervention. So what do we know? So if we look at prosthetics, so if you're setting a chip on the surface of the retina and you're stimulating cells that are 3 down from where photoreceptors should be, you're able shapes, movement, things like that. You don't the dynamics of it, the temporal sequence of it is off. 3 dimensions are hard to do because there's processing that goes on in the retina, and of course there's processing that goes on in the brain, ton in the brain. So from a photoreceptor, so an individual photoreceptor either detects light and releases chemicals or doesn't. Right? So it's the pinnacle of, or the first step in light detection. And so the further, it's a complex cell type, but the more you get to that, you start working with photoreceptors, the less you have to worry about the downstream processing, because you're letting what's there do the job. That's assuming that it's there and that it hooks up to what it should hook up to. So we know even in crude situations where you're bypassing photoreceptors and stimulating something down the stream, you still get useful vision. So for people who have no vision whatsoever, even knowing if the lights are on or off can help their sleep cycles, right? So there's all sorts of things that you get, and this is why back to the FDA question, right? I mean, there's a lot of things that people will say, you know what, I used to be able to detect light, and now I'm all screwed up, and I'm up in the middle of the night, and I'm sleeping during the day. And if they could have that back, their life would be better. So appropriately applying technology that we know works or we think might work for the purposes we want them to be used is important. So that brings up the idea. So the question I get more often is, okay, let's say you put that cell in and it reaches out, but what if it doesn't reach out to the thing it should reach out? It reaches out to the thing next to it, or misses it and goes forward and hits something. We don't know. So the question is, we assume that it would still be better than the prosthetics. How much better, we don't know. How many we have to have, how tightly they have to be packed. Because if you look at our retinas, the discrimination that we have has to do with how tightly packed one receptor for photons of light is next to the other one. That's why we're trying to pack more and more together. But at some point, you got to just fish and stop cutting bait, and you have to say, okay, what do we have here? Is there a signal? What's this? Where's our, what? Hopefully, what little thing works, or moderate thing, and what are the many things that don't? And then how do we fix it and move things forward, just like any other technology? So excellent questions, and depending upon where you want to intervene, there's cortical prosthetics now too, right? But things get so convoluted back in the brain that it's hard to know what to stimulate and how to put something together when you're dealing with something that's so far down the line. So as complex as all this is, from just a biological standpoint, the closer you are to the start, the easier, and the more you're relying on just what God already made, you're better off.
Hi, this question's for Dr. Rizzo. All the adaptive technology information is very exciting, and I'm kind of thinking about my high schooler who's going to be moving into college. And I'm wondering, how does this information that you're sharing with us today get disseminated to professional supports in those settings? And I'm thinking about the implications, especially with that shocking information about the disparity in, going to college and stuff.
It's a tough question. I think the best rule of thumb, and what I always start off with, I mean, I've had dozens and dozens of conversations, and the ophthalmologist that helped with me, even from when I was a young boy now, try to integrate me into conversations that they now have with young boys that are affected with both choroideremia and other retinal dystrophies, is advocate, advocate, advocate. So, if you have a child or someone in your family who's starting a new employment opportunity or is going to a new educational setting, in a lot of cases, people are very well intentioned, but they just don't know. Right. So you just tell them, hey, listen, these accommodations are unacceptable. Look what else exists out there. And then you have a pledge from me right now where I'm happy to send them education and evidence and, peer-reviewed publications. And we just continue to fire ammunition at them and say, listen, this is where the bar is, and the bar is a lot higher than you probably think. And we want more now. And this is what the potential is. And, can you start providing it for us? And if you continue to hammer away on it and at it, I think, we'll start to achieve big goals. But I really think it all comes down to you. I mean, your voices are the strongest voices, and I I like to think I'm a kind of a stubborn. I was raised by a lot of blue collar Italians in New Jersey, so you could read into that if you want. So I like to be polite up to a point, and then I lose my patience, and then I just call Uncle Paulie, and he takes care of it. No, I'm just kidding. He may or may not be in waste management, and may have gone away for a little while. That's somewhat true, but in all seriousness, I really think advocacy is really the key. And I'm happy to help to what extent I can in some of those discussions.
I'm going to use my voice. Okay. All right. Sorry. So this is the first of all, thank you for all the high school information because I called high school people and they are not aware for eyes things and so on and because we don't know exactly what the impact is yet in our kids. It's kind of a catch-22. So thank you for that. And that's a question for Dr. Gamm. So you showed us where you are in terms of all the technical advancement. And my question to you is, where are you in terms of, now you were talking about trials, where are you with that?
So yes, so we have a partnership with Bayer and Fujifilm and Blue Rock, and so the plans are for initial therapies. As you might imagine, replacing 2 cell types is more complex than one, and so the plan is to do one of them first, and then to do the 2 together. So as opposed to leaping in and doing one. So so choroideremia, which require 2, would not be first up, but would be in that next cohort, which still would be pretty, So first of all, not everything's gonna be perfect. So if everything goes perfectly, we're talking, within 5 years. But I will tell you, not everything will go perfect. But we have had multiple discussions now with the first product with the FDA, and they've gone well.
Sorry, the first trial.
Correct. Yeah. So photoreceptor replacement only. Yeah. Which would not be, we wouldn't do that for choroideremia if the RPE is already gone. But it would tell us that it's lots of things. Is the delivery correct? Is the formulation correct? Do we see a response? And the thing that we're always interested in is that even in RP, nothing is precise. So even though it's a photoreceptor disorder, just like if you have an RPE disorder, the photoreceptors die. If you have a photoreceptor disorder, the RPE doesn't need the photoreceptors, but they're not normal. So once they've been gone a long time, the RPE is also not quite. So we don't know. There's so many things we don't know. Like in a human, is the RPE still good enough? Because if you start doing things, sometimes will slough off. And so you're in the middle of a surgery putting in photoreceptors because you see the RPE is normal, and then the RPE starts to loosen up, and you're like, oh my goodness, I'm in the middle of, this is animal models, things like that. So these are all things that, so in the end, it may be that a dual therapy is best for everybody, right? Which would be kind of nice, because then you have one, and then you don't have to have a lot of multiple different things. But it's more complex. It's 2 cell types. It's a different delivery. The downside is we do it with a scaffold, which means that we have a smaller area that we can treat. So you rob Peter to pay Paul for almost everything. A great question.